अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 13 एक्सरसाइज लॉन्ग क्वेश्चन आंसर सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, डिस्क्राइब स्ट्रक्चर ऑफ क्रोमोसोम। अ क्रोमोसोम इज अ thread-like structure made up of DNA and protein, specifically histone protein, which carry genetic information. Components of chromosome are DNA, which is deoxyribonucleic acid, which is a double-stranded helix, histone, proteins which that DNA wrap around, and chromatin, complex of DNA and histone. Chromosome structure. Chromatid, single DNA molecule with associated histones. Sister chromatid, two identical chromatids joined at centromere. And centromere, region connecting sister chromatids. Chromosome regions. Telomeres, repetitive DNA sequences at chromosome ends. And centromere controls chromosome movement during cell division. Arms, long and short arms, P and Q arms. Chromatin organization. Nucleosomes, DNA wrapped around histone octamers. Chromatin fibers, coiled nucleosomes, loop domains. Chromatin fibers organized into loops. Next is chromosome types. Metacentric, in which centromere is present in the middle. Submetacentric, centromere is near the middle or slightly away from middle. Acrocentric, centromere near the end, and in telocentric, centromere is present at the end. Chromosome size and number. Humans have 23 pairs of chromosome and 46 in total, 3 billion base pairs, and chromosome length 50 to 250 million base pairs. Understanding chromosome structure is crucial for grasping genetics, inheritance, and cellular function. Next question is, describe in detail the composition and organization of chromosome. Chromosome composition and organization. First of all, chromosome structure. DNA, deoxyribonucleic acid, double-stranded helix carrying genetic information, histone protein, alkaline protein binding DNA forming chromatin, and non-histone protein, regulatory proteins, chromosomal scaffold. Next is chromatin organization. Nucleosome, basic unit, 147 base pair DNA wrapped around histone octamer. Chromatosome, nucleosome plus linker histone, H1. Solenoid model, nucleosomes coiled forming 30 nanometer fiber. Next is chromosome levels of organization. Interface, euchromatin active and less compact part, and heterochromatin inactive and compact part, more compact. Prophase, chromatin condense, visible chromosomes form, and in metaphase, chromosomes align at equator. Chromosome components, centromere, constricted region, attachment site for spindle fibers, telomeres, repetitive sequences TTA, GGG at chromosome ends, arms P short and Q long arms, chromosomes types 4 based on the position of centromere, metacentric, सबमेटासेंट्रिक, एक्रोसेंट्रिक एंड टीलोसेंट्रिक. मेटा में इक्वल आर्म्स होती हैं, सब मेटासेंट्रिक में अनइक्वल आर्म्स, एंड एक्रो में सेंट्रोमियर नियर एंड होता है, टीलो में सेंट्रोमियर एट एंड. Chromosome number and size. Humans have 23 pairs of chromosome and 46 chromosomes and 52 to 50 million base pairs. Chromosome functions, genetic information storage, inheritance, and gene expression regulation. Next question is, describe the experiment of Griffith to prove that DNA is hereditary material. Frederick Griffith experiment 1928. Objective क्या था इस experiment का? To investigate the transformative principle of bacteria. Materials क्या थे इस experiment के लिए? Two strains of Streptococcus pneumoniae, virulent pathogenic strain and non-virulent non-pathogenic strain type R. अच्छा, उसने क्या किया था? जो Streptococcus pneumoniae, उसके दो उसने स्ट्रेन्स लिए थे, एक एस टाइप और एक आर टाइप. ठीक है, जो हमारे पास था और माइस लिए, ठीक है। प्रोसीजर उसने क्या किया कि इंजेक्टेड माइस विद वायरुलेंट स्टेन माइस डाइट। उसने सबसे पहले किया कि जो माइस थे उनको इंजेक्ट किया वायरुलेंट स्ट्रेन से। तो माइस क्या हो गए, वायरुलेंट होते हैं डिजीज काजिंग, ठीक है। तो माइस डेड हो गए। फिर उसने जो नॉन वायलेंट थे टाइप आर वो इंजेक्ट किए, तो माइस लिव्ड। देन उसने हीट किल्ड वायरुलेंट स्ट्रेन इंजेक्ट किए, माइस अगेन लिव्ड। देन उसने क्या किया, मिक्सचर ऑफ हीट किल्ड वायरुलेंट एंड नॉन वायरुलेंट को मिक्स करके इंजेक्ट किया, तो माइस डाइड अगेन। ऑब्जर्वेशंस: mice injected with heat-killed virulent bacteria and non-virulent bacteria died. Bacteria isolated from dead mice showed presence of virulent stain. उन डेड माइस में ये देखा गया कि जो जिसमें उसने हीट किल्ड वायरुलेंट एंड जो नॉन वायरुलेंट इंजेक्ट किया था, तो उसमें उसने देखा कि जो वायरुलेंट लिविंग जो स्टेन है वो पाए गए थे। कंक्लूजन: उसने इस में कंक्लूड क्या किया, ग्रिफ्थ प्रपोज द कंसेप्ट ऑफ ट्रांसफॉर्मेशन के ट्रांसफॉर्मेशन होती है बैक्टीरिया में। नॉन-वायलेंट बैक्टीरिया एक्वायर्ड जेनेटिक मटेरियल फ्रॉम हीट किल्ड वायलेंट बैक्टीरिया। उसने कहा कि जो नॉन वायलेंट बैक्टीरिया थे उन्होंने जेनेटिक मटेरियल ले लिया हीट किल्ड वायलेंट बैक्टीरिया से। दिस जेनेटिक मटेरियल ट्रांसफॉर्म्ड नॉन वायलेंट इनटू वायलेंट बैक्टीरिया। लेटर एवरी मैकलॉड एंड मैकार्टे 1944 identified DNA as transformative principle. Key findings: DNA is responsible for bacterial transformation and DNA carries genetic information. Impact: Griffith experiment laid the foundation for identifying DNA as genetic material, understanding bacterial transformation, and development of molecular biology. Limitations: Griffith didn't isolate DNA, but his experiment paved the way for future research. Next question is: Describe the experiment of Hershey and Chase to prove DNA is hereditary material. The Hershey and Chase experiment, which was performed in 1952, was a landmark study confirming DNA as genetic material. अच्छा बच्चों, 1952 से पहले ये पता नहीं था कि जो हेरिडिटरी मटेरियल है, जो इन्फॉर्मेशन वन जनरेशन नेक्स्ट लेके वो कौन सा है। इधर इट वाज डीएनए और प्रोटीन, क्योंकि प्रोटीन्स में जो है हमारी बॉडी में बहुत ज्यादा फंक्शंस परफॉर्म कर रहे थे, लाइक एंजाइम्स, प्रोटीन्स हैं, ठीक है, डिफरेंट। तो इधर इस कंफ्यूजन बिटवीन प्रोटीन एंड डीएनए के हेरिडिटरी मटेरियल कौन सा है। सो हर्शी एंड चेस ने 1952 में एक्सपेरिमेंट किया और प्रूफ किया कि जो हेरिडिटरी मटेरियल है वो डीएनए है, ठीक है। सो ऑब्जेक्टिव टू डिटरमाइन वेदर डीएनए और प्रोटीन इज रिस्पांसिबल फॉर जेनेटिक इनहेरिटेंस। ओके, मेथोडोलॉजी क्या थी इनकी, बैक्टीरियोफेज T2 वायरस इन्फेक्टिंग यूकोलाई वाज यूज्ड। अच्छा, बैक्टीरियोफेज बच्चों ऐसे वायरस, फेज कहते हैं वायरस को, बैक्टीरियो फ्रॉम बैक्टीरियम, दोज वायरसेस व्हिच इन्फेक्ट बैक्टीरिया, उनको हम बैक्टीरियोफेज कहते हैं। इन्होंने बैक्टीरियोफेज वायरसेस लिए, टू सेपरेट एक्सपेरिमेंट इन्होंने कंडक्ट किए। फर्स्ट वन इज हर्शीज एक्सपेरिमेंट डीएनए लेबलिंग। इन्होंने पहले डीएनए को लेबल किया। रेडियो लेबल्ड फास्फोरस P32 incorporated into DNA and infected E. coli with labeled phages. अच्छा, हर्शीज एक्सपेरिमेंट क्या किया, उसने डीएनए को लेबल किया। उसने क्या किया कि डीएनए को लेबल किया फास्फोरस 32 से और उसको इन्फेक्ट करवाया ई कोलाई को, ठीक है। देन चेज एक्सपेरिमेंट क्या, प्रोटीन, उसने प्रोटीन लेबलिंग की। उसने क्या किया, जो प्रोटीन थी उसको लेबल किया सल्फर 35 से और ई कोलाई को इन्फेक्ट करवाया, ठीक है। Infected cells were sheared, releasing phage codes. Centrifugation separated phage code from infected cell. Results क्या है हमारे पास, डीएनए लेबलिंग, radioactivity found in infected cell, DNA entered cells transforming genetic material. And protein labeling, radioactivity remained in phage code, protein did not enter cells. Conclusion: DNA, not protein, is the genetic material. Key findings: DNA is responsible for genetic inheritance. DNA enter host cell during infection, and protein plays a structural role. Impact: confirmed Avery's findings 1944 on DNA's role and established DNA as the central molecule of genetics. Influenced Watson and Crick DNA structure discovery in 1953. Limitations: Used bacteriophages, not cellular-organisms, assumed phage DNA represented on DNA. The Ashish Chase experiment provided crucial evidence for DNA's role in genetic inheritance. Next question is, describe the three models proposed about mechanism of DNA replication. The three models were conservative, semi-conservative and dispersive model. We will discuss each one in detail. First one is conservative model proposed by Watson and Crick. Key features of this model are parental DNA remain intact, new DNA synthesized from parental template, no mixing of old and new DNA. Limitations not supported by experimental evidence, does not explain semi-conservative nature of replication. Next is dispersive model proposed by Taylor and others. Key features: parental DNA broken into fragment, new DNA synthesized from fragmented template, mixing of old and new DNA. Limitations not supported by experimental evidence, does not explain semi-conservative nature of replication. Last one is semi-conservative model, proposed by Watson and Crick, confirmed by Michelson and Stahl experiment in 1958. Key features: parental DNA unwind, separate into two strands. Each strand serves as template for new DNA synthesis, resulting DNA one old and one new strand. Semi-conservative. Evidence: density gradient centrifugation experiments by Michelson and Stahl. अच्छा बच्चों, ये जो तीन मॉडल ऑफ रेप्लिकेशन है ना, कंजर्वेटिव, सेमी कंजर्वेटिव और डिस्पर्सिव, इन तीनों ने कहा था कि रेप्लिकेशन इस मॉडल्स के अकॉर्डिंग होती है। लेकिन बाद में जो मिजल्सन एंड स्टाल है, इन्होंने एक्सपेरिमेंट करके 1958 में ये प्रूव किया था कि जो डीएनए की रेप्लिकेशन है वो थ्रू सेमी कंजर्वेटिव मॉडल होती है। ठीक है, कंसिस्टेंट विद जेनेटिक इनहेरिटेंस पैटर्न। द सेमी कंजर्वेटिव मॉडल is widely accepted as accurate mechanism of DNA replication. Key enzymes involved are helicase, which unwind DNA double helix, primase, which synthesizes RNA primer, DNA polymerase, which synthesizes new DNA strand, and ligase, it sees gap between Okazaki fragments. Next is application process. It consists of following steps. First one is initiation, then unwinding, then senses of leading and lagging strand, then proofreading and editing, and then completion. Next question is describe work of Michelson and Stahl to justify semi-conservative replication as correct method of replication. अच्छा, हमने ऊपर पढ़े ना कि रेप्लिकेशन के तीन मॉडल्स प्रपोज किए थे, कंजर्वेटिव, सेमी कंजर्वेटिव एंड डिस्पर्सिव। तो कह रहे हैं कि मिजल्सन ने स्टाल का वर्क एक्सपेरिमेंट बताएं जिसमें उन्होंने प्रूव किया था कि जो डीएनए की रेप्लिकेशन है वो सेमी कंजर्वेटिव मॉडल के थ्रू होती है। सो मिजल्सन ने स्टाल एक्सपेरिमेंट परफॉर्म्ड इन 1958। इनका ऑब्जेक्टिव क्या था एक्सपेरिमेंट का? टू डिटरमाइन मैकेनिज्म ऑफ डीएनए रेप्लिकेशन। हाइपोथेसिस: कंजर्वेटिव रेप्लिकेशन, पेरेंटल डीएनए रिमेन इंटैक्ट, न्यू डीएनए सिंथेसाइज। तो हाइपोथेसिस ये थी कि अगर कंजर्वेटिव रेप्लिकेशन होती है तो जो पेरेंटल डीएनए वो एग्जैक्ट सेम रहना चाहिए, इंटैक्ट रहना चाहिए, और साथ में न्यू डीएनए सिंथेसाइज होना चाहिए। डिस्पर्सिव क्या रेप्लिकेशन क्या है कि पेरेंटल डीएनए फ्रेगमेंट में टूट जाए और फिर मिक्स हो जाए न्यू डीएनए के साथ। और सेमी कंजर्वेटिव ये था कि पेरेंटल डीएनए स्टैंड सेपरेटेड, ईच पेयर्ड विद न्यू डीएनए। ओके, इन्होंने मेथोडोलॉजी क्या उससे यूज की? ई कोलाई कल्चर्ड इन हैवी नाइट्रोजन मीडियम, cells transferred to light nitrogen medium, DNA isolated and centrifuged in cesium chloride density gradient. ओके, इन्होंने क्या किया कि जो ई कोलाई है उसको क्या कल्चर करवाया हैवी नाइट्रोजन मीडियम पे, देन उनको ट्रांसफर किया नाइट्रोजन 14 मीडियम पे। ठीक है, फिर उन्होंने डीएनए आइसोलेट किया एट 0, 20 एंड 40 मिनट्स, एंड देन क्या किया, उसको सेंट्रीफ्यूज किया और डेंसिटी को चेक किया। रिजल्ट्स क्या आए? जो हमारे पास जो फर्स्ट जनरेशन थी डीएनए आफ्टर वन एप्लीकेशन, जिसकी जो वो डेंसिटी थी हाइब्रिड थी, यानी बिटवीन नाइट्रोजन 15 एंड नाइट्रोजन 14 स्ट्रैंड्स के बीच में थी। देन सेकंड जनरेशन डीएनए आफ्टर टू रेप्लिकेशंस, टू डिस्टिंक्ट बैंड्स आए, एक लाइट 14 नाइट्रोजन 14 बैंड आया और एक हाइब्रिड बैंड आया। कंक्लूजन क्या हुई इससे हमारे पास कि सेमी कंजर्वेटिव रेप्लिकेशन कंफर्म। की फाइंडिंग्स: parental DNA strand separate during replication, each parental strand pair with newly synthesized DNA, and new DNA synthesis occur in semi-conservative manner. इसने कहा था कि पेरेंटल डीएनए पहले तो वो सेपरेट हो जाएगा, तो उसके दोनों स्ट्रैंड, हर स्ट्रैंड के सामने एक न्यू स्ट्रैंड बनेगा, और जो न्यू डीएनए बनेगा उसमें एक पेरेंटल स्ट्रैंड होगा और एक न्यूली सिंथेसाइज्ड डीएनए होगा। इस तरह जो रेप्लिकेशन हुई है वो सेमी कंजर्वेटिव हुई है। ठीक है। इम्पैक्ट, confirmed Watson and Crick replication model of 1953 established semi-conservation replication as universal mechanism, supported double helix DNA of model of DNA. Experimental design, density gradient centrifugation and nitrogen isotopes to differentiate DNA and limitations. Use prokaryotic cells and assume DNA replication mechanisms are universal. Next question is, describe event of process of DNA replication and explain DNA stability and variability as two factors of replicating DNA molecule. ओके, हमने जो डीएनए रेप्लिकेशन के स्टेप्स है वो बताने हैं। तो डीएनए रेप्लिकेशन का जो सबसे पहला प्रोसेस है वो इनिशिएशन होती है। पहले के सबसे पहले क्या होगा, जो हेलिकेज एंजाइम है वो डीएनए डबल हेलिक्स को अनवाइंड कर देता है, और किस पोजीशन से जहां से करता है उसको ओरिजिन ऑफ रेप्लिकेशन ओआरआई कहते हैं। देन प्राइमेज जो है एंजाइम, वो क्या करेगा, आरएनए प्राइमर्स को सिंथेसाइज करेगा टेम्पलेट स्टैंड के ऊपर। नेक्स्ट स्टेप इज अनवाइंडिंग। टोपोआइसोमेरेज जो है ना वो रिलैक्स कर देगा सुपर कॉइल डीएनए को, और जो हेलिकेज है वो कंटिन्यूस करेगा अनवाइंडिंग। देन होगी सिंथेसिस। डीएनए पॉलीमरेज रीड टेम्पलेट स्ट्रैंड एंड मैचेस बेसिस। लीडिंग स्ट्रैंड कंटिन्यूस सिंथेसिस होगी और लैगिंग स्ट्रैंड डिसकंटिन्यूस ओकर की फ्रेगमेंट्स। आरएनए प्राइमर रिमूव्ड रिप्लेस्ड विद डीएनए। देन होगी हमारे पास प्रूफ रीडिंग एंड एडिटिंग। डीएनए पॉलीमरेज करेक्ट एरर, प्रूफ रीडिंग एक्सोन्यूक्लियस रिमूव इनकरेक्ट बेसिस। Then completion, ligase joins the gap between key fragments and replication fork merged. DNA stability and variability. Stability characteristics, high fidelity, accurate base pairing, proofreading and editing mechanism, DNA repair pathways, which include mismatch repair, nucleotide excision repair, and then telomere maintenance. Variability characters, these include mutation, errors during replication, recombination, crossing over during meiosis, gene duplication, copy number variation, and epigenetic modifications, gene expression regulation. Factors influencing DNA stability and variability. Environmental factors which include radiation, chemicals, etc. Genetic factors include mutation and repair efficiency. Cellular processes include stress or telomere shortening. Consequences of alter DNA stability and variability. Genetic disorders, cancer, aging, and evolutionary changes. Next question is, describe mechanism of polymerase 3 activity. Polymerase 3 is primary enzyme responsible for DNA replication in bacteria. Its mechanism involve initiation, binding. Polymerase 3 holoenzyme bind to template DNA at replication fork. Primer recognition. Polymerase 3 recognizes RNA primers synthesized by primase assembly. Polymerase 3 subunits alpha, beta, gamma, epsilon assemble around the template. Next is elongation. Nucleotide incorporation. Polymerase 3 matches incoming nucleotides to the template DNA. Phosphodiester bond formation. Polymerase 3 forms phosphodiester bond between nucleotide and processivity. Polymerase 3 beta clamp ensure continuous synthesis, allowing for rapid elongation, 700 to 1000 nucleotide per second. Next is proofreading and editing. Exonuclease activity. Polymerase 3 epsilon subunit removes incorrect nucleotides. And correction. Polymerase 3 replaces incorrect nucleotide with correct one. Then termination. Completion. Polymerase 3 reaches the end of template DNA. Release. Polymerase 3 releases the newly synthesized DNA strand. Key features: high processivity, long DNA synthesis without dissociation, high accuracy rate, and rapid synthesis rate. Subunit roles: alpha catalytic subunit, epsilon proofreading subunit, theta stabilizes epsilon subunit, beta sliding clamp processivity, and dimerizes alpha subunit. Next question is explain the mechanism of transcription. Transcription is process of synthesizing RNA from DNA. It consists of first initiation phase. RNA polymerase bind to promoter region. Sigma factor facilitate binding. And DNA unwind forming transcription bubble. Then होगी elongation. RNA polymerase read DNA template. Nucleotide complementary to template added to growing RNA chain. And RNA senses occur in 5 to 3' direction. Then termination. RNA polymerase reaches termination sequence. Hairpin loop forms in RNA and RNA released polymerase detaches. Post-transcriptional modifications include splicing, introns removed, exons joined, capping, 5' ethyl guanosine cap added, and polydenylation, 3' poly A tail added. Types of RNA, 3 types: messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA protein synthesis, transfer RNA transport amino acid, ribosomal RNA component of ribosome. Regulation of transcription, promoters, enhance or inhibit transcription, enhancers, increase transcription efficiency, and repressors decrease transcription. Transcription factors, general transcription factors which play a role in initiation, and specific transcription factors regulation. Mechanism of transcription in eukaryotes, RNA polymerase II transcribe messenger RNA, mediator complex facilitate polymerase II binding, and histone modification regulate chromatin accessibility. Next question is describe the mechanism of protein synthesis, which is also known as translation. It consists of first step transcription. During transcription, factors bind to promoter region. DNA polymerase synthesizes messenger RNA, and messenger RNA undergoes processing like splicing, capping, and polydenylation. Next step is translation initiation. Ribosome bind to messenger RNA at start codon. Initiation transfer RNA binds to start codon, and initiation factors facilitate ribosome assembly. Then elongation. Ribosome read messenger RNA sequence in triplet codon. Transfer RNA molecules bring corresponding amino acid. Peptide bond form between amino acid. Ribosomes move along messenger RNA elongating polypeptide chain. Next step is termination. Ribosome reach stop codon like UA, UAG, UGA. Release factors facilitate termination. Completed polypeptide chain release. Mechanism detail. Codon recognition, transfer RNA anticodon pairs with messenger RNA codon. Amino acid activation. Amino acid transfer RNA synthetase link amino acid to transfer RNA and peptide bond formation. Ribosome catalyzes peptide bond. Key players, ribosomes, cellular machinery for translation, transfer RNA, adapter molecules linking amino acid to codon, amino acid transfer RNA synthetases, enzymes linking amino acid to transfer RNA, initiation and elongation factors, proteins facilitating translation. Post-translational modifications include proteolytic processing, folding and chopping, glycosylation, and phosphorylation. Next question is write the difference between protein synthesis in prokaryote and eukaryote. Initiation. In prokaryotes, Shine Delgado sequence, ribosome binding site, small subunit 30S bind messenger RNA, initiator transfer RNA binds start codon AUG, and larger subunit 50S joins. And in eukaryotes, initiation process involves cap binding complex which recognizes messenger RNA, small subunits 40S binds messenger RNA, initiator transfer RNA binds start codon, and largest subunit 60th join. Then translation in prokaryotes. Polycistronic messenger RNA, multiple genes and no nuclear membrane, simultaneous transcription and translation के साथ साथ होती है and 70S ribosomes are involved. While in eukaryotes, monocistronic messenger RNA and single gene nuclear membrane separate transcription and translation. Post-transcriptional modifications like splicing and capping and ATS ribosomes are involved. Next is elongation. Prokaryotes, faster elongation rate, 20 amino acids per second and fewer initiation factors, while in eukaryotes, slower elongation rate, 2 to 5 amino acids per second, more initiation factors. Next is termination. Release fact in prokaryotes, release factors RF1, RF2 recognizes stop codon and ribosome recycling factor RF, while in eukaryotes, release factors recognizes stop codon and ribosome recycling factor. Post-transcriptional modifications in prokaryotes, limited modification, no membrane-bound organelles, while in eukaryotes, extensive modifications like phosphorylation, glycosylation, membrane-bound organelles. Regulation. In prokaryotes, transcriptional regulation dominate, fewer post-transcriptional regulation, while in eukaryote, post-transcription regulations prominent, microRNAs regulate gene expression. Next question is, what is the importance of regulation of gene expression? Regulation of gene expression is crucial for cellular differentiation and development, tissue-specific gene expression, response to environmental changes, adaptation to stress, maintenance of cellular homeostasis, prevention of genetic disorder, cancer prevention, immune system function, nervous system development and function, overall organismal health. Consequences of dysregulated gene expression are genetic disorder, cancer, neurological disorder, immunological disorder, and developmental abnormalities, metabolic disorders, and aging. Mechanism of gene expression regulation. Transcriptional regulation like enhancers and promoters. Post-transcriptional regulation, epigenetic regulation, DNA methylation and histone modification. Translational regulation, messenger RNA stability and ribosome binding. And post-translational regulation, which include protein modification. Next is regulation of gene expression enables cells to respond to signal, adapt to changing condition, maintain cellular identity, prevent unnecessary gene expression, optimize source allocation. In summary, regulation of gene expression is essential for maintaining cellular homeostasis, ensuring development and preventing diseases. Next question is, describe negative control of gene expression by repressor protein and the positive control by activator protein. Negative control of gene expression by repressor protein. Mechanism. Repressor protein bind to operator region, blocks RNA polymerase binding to promoter, and prevent transcription initiation. Key components include repressor protein, operator region, and promoter region. Examples include Lacopron E. coli. Lac oppressor protein binds to operator, blocks RNA polymerase binding to promoter, prevent transcription of lac gene. Regulation. Inducer molecules bind repressor conformation changes, release operator, RNA polymerase bind, promoter transcription initiate. Next is positive control by activator protein. Mechanism. Activator protein bind to enhancer region, recruits RNA polymerase to promoter, enhance transcription initiation. Key components include activator protein, enhancer region, and promoter region. Examples are GAL4 in yeast. GAL4 activator protein bind to upstream activating sequence and recruits RNA polymerase to promoter, enhance transcription of GAL genes. Regulation. Activator protein bind to enhancer, routing coactivators, chromatin remodeling, enhances RNA polymerase binding transcription. Comparison. Negative control repressor, default state transcription off, regulation, repressor binding, prevent transcription. Positive control or activator, default state transcription off, regulation, activator binding enhances transcription. Next question is, explain post transcription modifications of messenger RNA. First one is, define mutation and identify various sources of mutation. Differentiate between natural and induced mutation. Explain harmful and useful aspect of mutation. And next is, rationalize mutations might be a contributing factor toward evolution. Post-translational modifications of messenger RNA. These are changes made to messenger RNA after transcription from DNA. These regulate gene expression, messenger RNA stability, and protein synthesis. These include 5' capping, which add a methylated guanine nucleotide to protect messenger RNA from degradation. Next is polyadenylation. Adds adenine nucleotide to stabilize messenger RNA. Next, splicing, which remove intron and join exon, and editing, change individual nucleotides, and methylation, add methyl group to regulate translation. Next is mutation, sources and type. Mutation is a permanent change in DNA sequence. Sources are error during replication and environmental factors like radiations, chemicals, etc., genetic recombination during meiosis, mobile genetic elements like transposons, and DNA repair errors. Types of mutations are point mutation, which is single nucleotide change, then frame shift mutation, chromosomal mutations, and genome mutations. Next is natural versus induced mutations. Natural mutations which occur spontaneously or through errors during DNA replication. Induced mutations result from external factors like radiations, chemicals, viruses, etc. Next is harmful and useful aspect of mutation. Harmful: genetic disorders like sickle cell anemia, cancer, and reduced fitness. Useful like variation and diversity, adaptation to changing environment, and evolutionary innovation. Mutations and evolution. Mutation provide the raw material for evolution. They introduce new traits, increase genetic diversity, allow population to adapt, and drive speciation. Next question is describe symptom, causes, and possible treatment of following chromosomal mutation: Down syndrome, Klinefelter syndrome, and Turner syndrome. First one is Down syndrome. Symptoms include intellectual disability, delayed speech and language development, and their physical characteristics like flat face, small ears, and congenital heart defects, increased risk of respiratory problems, vision impairment, and hearing loss. Causes trisomy 21, extra copy of chromosome 21, and translocation, part of chromosome 21 attaches to another chromosome. Treatment: early intervention programs like speech, physical, occupational therapy, special education and sport, medical management, heart surgery, vision correction, lifestyle modification, healthy diet. Klinefelter syndrome. Symptoms include trust issues and learning disabilities and emotional and behavior issues. Causes: an extra X chromosome and genetic mutation. Treatment: hormone replacement therapy, speech and language therapy, and physiological counseling. Next is Turner syndrome. Symptoms include saturature, heart defects, kidney problem, hearing loss, learning disability and cognitive impairment, emotional and behavioral issues. Causes: monosomy, missing one chromosome, genetic mutation. Treatment: hormone replacement therapy, growth hormone therapy, heart surgery, speech and language therapy, psychological counseling. Next question is describe the symptoms, causes, and possible treatment of following genetic mutation: sickle cell anemia and phenylketonuria. Sickle cell anemia. Symptoms include recurring episode of severe pain, anemia, fatigue, shortness of breath, increased risk of infection, jaundice, yellowing of skin and eyes, organ damage, kidneys, liver, heart, etc. Causes: point mutation in HBB gene, beta globin subunit, and substitution of glutamic acid with valine, and autosomal recessive inheritance. Treatment: blood transfusion, pain management, antibiotics, vaccination, hydroxyurea, and stem cell transplantation and gene therapy. Next is phenylketonuria. Symptoms include intellectual disabilities, seizures, behavioral problems, delayed speech and language development, pale skin, hair loss, and causes include point mutation in PAH gene, phenylalanine hydroxylase, deficiency of phenylalanine hydroxylase enzyme, and autosomal recessive inheritance. Treatment include dietary restriction of phenylalanine, low protein diet, phenylalanine free formula, supplements like tyrosine, vitamin B12, regular blood test, phenylalanine levels, gene therapy experimental. Newborn screening. Both sickle cell anemia and PQ are included in newborn screening programs allowing for early diagnosis and treatment. Prenatal testing available for both conditions. enabling parents to make informed decisions. Genetic counseling is essential for families with a history of these conditions, providing information on risks, inheritance and testing options. तो जो हमारे इलाकों से कंप्लीट हो चुके हैं, होपफुली आपको समझ आ गए होंगे. इफ यू हैव एनी डाउट यू कैन आस्क इन कमेंट सेक्शन. प्लीज लाइक शेयर एंड सब्सक्राइब माय यूट्यूब चैनल. थैंक यू.